Loon is a visualization technique to analyze large-scale microscopy data using exemplar data points. The ultimate goal of our collaborators is to improve cancer treatment. Cancer occurs when cells grow and divide rapidly. Unfortunately, cancer is highly heterogeneous, meaning that different kinds of cancer behave quite differently. To combat this, many different drugs have been developed. However, it is difficult to know which drugs will be effective for a particular patient. Our collaborators use a quantitative phase microscope to measure the growth of cancer cells that have been treated with different drugs in order to find an answer to this question. Quantitative phase microscopes measure how much light slows down as it passes through cells. This can be used to derive the mass of cells. To measure many different experimental conditions at once, our collaborators place cells on a plate with many wells and expose each well to different drugs at different concentrations. Here, color represents these different drugs. They then take images at multiple locations within each well. For each image, our collaborators segment the cells from the background. Next, these cell segments are tracked over time by connecting regions with similar positions and mass. These tracks are saved along with derived cell attributes such as the mass or area of a cell. This information can then in turn be used to compute track attributes, such as the growth rate of a cell. An effective cancer drug for a specific patient inhibits growth at a low concentration. Loon provides multiple views to give insight into this microscopy data. We can select experimental conditions to remove those that don't seem to respond to drugs, and we can then compare these conditions using aggregate growth curves. The image selection view lets us choose specific images which are displayed in the image view. Aggregate cell and track attributes are shown as histograms and scatter plots, which can be brushed to select data. Finally, the exemplar tracks view visualizes representative cells. Now, let's take a closer look at some of these views. The mass of multiple cells can be averaged over time to get a single growth curve. Here, we facet the data based on experimental conditions. Each row is a different drug, and each column is a different concentration. In this case, we have 5 drugs and 2 controls. Let's filter our data to show every other concentration, which is reflected in the entire interface. For a more detailed comparison, we can also superimpose the growth curves in a single line chart. In addition to growth, we can view how different dimensions behave over time. In this case, we are looking at the density of cells. Given the large number of images taken, we need means to efficiently navigate them, which we achieve with the image selection view. Each row in this view corresponds to an imaging location. The rows are grouped by condition. Each tick mark in this row represents a single image. The size of the tick mark encodes the number of selected cells at each frame. Similarly, the bar encodes the number of selected cells at the entire location. A tick mark can be selected to show the image at that frame. The image viewer shows both the images and the segmented contours of the cell, but can be toggled off to help focus on the relevant cells. Keyboard controls can be used to quickly animate cell growth over time. While it is possible to inspect some cells for every condition with this view, it can be quite tedious. To remedy this, we introduced the Exemplar Tracks view, which automatically extracts cell tracks from every condition. Here is how extracting exemplars works. We begin with cell tracks that are exposed to different drugs. First, we group these tracks based on the drug. Then, within each group, we sort by an attribute of the cell track, such as its average mass. We then strategically sample specific tracks to display as exemplars. When aligned strictly by time, these tracks can be very long. To address this, we also sample individual cells over time. In addition, we keep track of the lifetime of the cell within the experiment and show it as a bar above the exemplar. This way, we can realign the tracks while preserving the temporal context of the cell track. This is how this looks in Loon. Here, we show 2 additional charts. The distribution of mass for the current drug is shown in a histogram on the left. Pins mark where the exemplars are sampled from. The growth curve on the right shows the exemplar's mass over time, as well as the average curve for that drug. Let's take a look at this in action. We begin with exemplars selected based on their average mass at the 5th percentile, median, and 95th percentile. The sampling is repeated for every experimental condition. Currently, we have 3 drugs selected, each with a concentration of 20 micromolars. If we see a particularly interesting cell, we can select it to view the image it was extracted from. Furthermore, I can bookmark cells to add them to the exemplar tracks view. To get exemplars of different shapes, I can use the shape factor instead of size to sample data. Loon provides several pre-specified sampling strategies, but we can also manually pick a specific value from the distribution for which an exemplar is extracted. The following is an illustration of how Loon was used by our collaborators in a case study. In this dataset, we are trying to understand the effect of different drugs on the cells. We began by viewing the condition selection matrix. Here, we see a sharp decline in the growth rate between these two concentrations. This would indicate that cells are dying at this higher concentration. We select this condition to confirm that cells truly are dying. Next, we retrieved exemplars based on the growth rate. To differentiate dying cells, we select only tracks with a negative growth rate. To see additional dying cells, we add a new pin on the distribution. In this cell, we see a large decrease in mass over time. However, we also see that this is in part due to a cell leaving the segmented region. We can examine other cells by adjusting the manual pin value. Here, we see a clearer example of a cell dying. Bookmarking this cell makes it easy to reference in later comparison and communication. This combination of cell growth curves and manual inspection of images is instrumental in verifying that these cells truly are dying, and the processing of the data reflects this phenomenon. We hope you liked our tour of Loon. Thank you for watching our video.